Hi, it's Glenn Wokenfeld, Mr. W from learn-biology.com or sciencemusicvideos.com. This video is called Understanding Genetic Engineering. It's part of the Amgen Biotech experience made by myself and my colleagues at Berkeley High School. And it's an RFP, red fluorescent protein transformation lab. And let's start by talking a little bit about the Amgen Biotech experience. This is a program that's put forward by the company Amgen, which provides teachers with fantastic professional development in biotechnology so that they can implement a series of labs with their students. Amgen provides all of the materials for free. And so it's great professional development. It's a great experience for your students. I can't recommend it highly enough. Go to the web address that's on the screen and see if you can enroll. It's in many parts of the country, and again, I can't recommend it highly enough. So let's talk about genetic engineering. Since the late 1970s, so that would be about 40 years, humans have been able to do a new thing on this earth, and that is they've been able to go into the nucleus of a eukaryotic cell, and that of course includes a human cell, and they've been able to extract genes. They've been able to go into bacterial cells and extract tiny circles of DNA called plasmids, circles of DNA that are outside the main bacterial chromosome. And they've been able to use enzymes to cut open these plasmids and then insert the human gene into that plasmid. And what does that give you? That gives you an engineered plasmid. And that's called recombinant DNA. It's DNA from different sources that's been recombined into one strand. And that has happened forever within one species. But to have that happen between species, that's genetic engineering. Well, you can take that plasmid and you can insert it into a bacterial cell. And that's what you have over here. And then you can grow those bacterial cells in large cultures, and you know, DNA makes RNA makes protein, and so you can extract the protein, and that's what you have over here. It's a eukaryotic protein that's produced by an engineered bacterium. Is that important? You bet it is. So you probably know that the disease diabetes is often caused by a person's inability to produce the hormone insulin, which controls blood sugar levels and causes cells to take up that sugar and convert it into glycogen or fats or so on and so forth. So that was figured out about 1900, and for the subsequent 100 years, the only source of insulin was from, our relatively close cousins, the mammals. So sheep, pigs, cows, goats. And basically what you'd have to do is you'd have to extract the pancreas from these animals after they were slaughtered for food, and then you could extract the insulin from the pancreas. That worked pretty well, because we're all mammals, these molecules are very close to one another. But for the past 20 years or so, humans have been able to make insulin that's actual human insulin, amino acid for amino acid, the same molecule, the same protein. And that has many benefits. And again, this is only one of many gene products that are being created with the technology that we're going to learn about in this series of labs. So the goal of this lab is, well, severalfold. There are some learning goals and then there's sort of a technical goal. So one thing that we're going for is this over here. This is a plate of bacteria, and these bacteria are E. coli bacteria, the kind that grow in our guts, but they've been genetically engineered. And so what they've experienced is what's called transformation. They've been forced to take up a plasmid, and that plasmid contains the red fluorescent protein. That's originally a eukaryotic protein found in sea anemone, and so that gene was put into a plasmid. We're going to put that plasmid into bacterial cells. If the lab works, we're going to get it to produce this red color that actually fluoresces under fluorescent light. I'll show you that a little bit later. Now, to achieve our goal, we're going to do a couple of other things. So to begin with, first of all, we're going to digest, cut open this plasmid. Now, this is actually the plasmid that contains the red fluorescent protein gene. And this part of the lab is more sort of a proof of concept. And it's to show you that plasmids can be cut open. And the parts that make up the plasmid, the segments that make up the plasmid can be analyzed. And so the next thing that we're going to do is we're going to verify that it's the plasmid that we want. And then we're going to take this plasmid, insert it into a cell, and then finally we're going to grow and screen for the transformed bacteria to make sure that we've got just the bacteria that we want. And just to learn the lingo, the first step is called restriction. It uses restriction enzymes to cut open the plasmid. The second step is called electrophoresis. It's using an electrical current to push molecules around. The third is called transformation, and there's no special name for the fourth, growing and screening bacteria. But remember those other three terms. Okay, so I am really lucky to work with a fabulous team of teachers at Berkeley High School. And the teachers who you're going to see actually performing the steps of this lab are my three wonderful colleagues, Colleen Simon O'Neill, Johanna Berg, and Sydney Ardal. They'll be carrying out and explaining various portions of the lab. I am one lucky teacher to work with such fabulous colleagues. So let's start by talking about some tools of the trade. So what you see in, I believe, Colleen's hand is a micropipette. So when we're doing biotechnology, we're moving incredibly small volumes of liquid around. So like, for example, a microliter is 1/millionth of a liter. Microliter is 1/millionth of a liter. So you know what a liter bottle looks like. So 1/millionth of that, that's a tiny quantity of liquid. You can pick that up. You can move it around. And so that's one thing that happens in this lab, and it happens with this device. That's called a micropipette. You also need to sort of mix these liquids and actually spin them down, and you do that using what's called a centrifuge, a microcentrifuge. So these are actually little microtubes. They hold small quantities of liquid, and you can move them around. And this is an electrophoresis chamber. That's by far the most technical, and biologically speaking, in terms of AP biology and our learning goals, by far the most important part. So let's talk about electrophoresis and how it works. Electrophoresis means pushing with electricity. And what it is a technique that allows us to separate molecules by size and charge. And what do I mean by charge? Just think about what you know about molecules. Molecules have functional groups attached to them. So for example, a phosphate group is a negatively charged molecule. So that's one example of a charge, and using charges like that, you can move molecules around. So what we see here is a micropipette. This is the tip of a micropipette, and this is a gel. It's made of a substance called agarose. That's a seaweed extract. Inside the gel is a kind of buffer, and this whole thing is in the chamber that I showed you before that an electrical current can pass through. That's why there's a negative sign over here for a negative charge and a positive sign over here for a positive charge. So you put the molecules, or whatever it is you're testing, that can be DNA, it can be ink, and you put it in over here, you turn on the current, and then what happens is that the electricity actually moves the molecules across. So you can, again, separate molecules by size and charge. And what we're going to see now is Colleen go ahead and do that. So let's get this to happen. Here it is. So Colleen is putting a tiny bit of ink into this. Oh, she's pulling it out of that microfuge tube. And what she's doing is she's now loading ink into the three wells, and one of the three wells of this electrophoresis setup. And I just want to point out that she's previously done that over here and over here. We're just showing the last one. This is the tip of the micropipette, and so now it's set to go. So what Colleen is now going to do is she's going to turn the machine on, and let's see that happen here. So once it's on, remember there's a negative electrode over here, there's a positive electrode here, and let's watch. So this is time-lapse photography. And what you can see is, in this case, it was ink, is actually getting pushed across the gel underneath the liquid. It's being separated out, and you can see that some of these inks actually have component colors that make them up. And so here it is. We're running to the end of the gel, and we're going to stop it. And now on the next slide, what we'll do is we will take a closer look. So now you can see that these three inks, here was the well over here. You can see that one had these two component colors. This one had three component colors. Looks like this one only had one over here. Again, the main thing to remember is that electrophoresis enables you to separate molecules by size and electrical charge. And so what that means is that if you're separating DNA, you can separate DNA fragments by size and electrical charge. And we'll see that done in this lab. So the first part of this lab involves digestion followed by verification of a plasmid. So this is our genetically engineered plasma, P-ARA-R, and it's a plasmid that is exactly 5,302 base pairs. It's a circular plasmid, so the beginning and the end circle up together. This is engineered. Here's the red fluorescent protein gene that's inside here. Here's a gene for ampicillin resistance, and here's a gene for gene control element that we'll be talking about later. So why are we doing this? One thing is that we want to demonstrate the techniques that we use to build plasmids like this. We cut them open, and using other enzymes we can put them together. And it's also how we verify a plasmid, because a plasmid, it's a piece of DNA, it makes RNA, it makes protein. You can test it by actually putting it inside a cell and seeing what it makes, but there are much easier ways that you can do it without growing a cell. You can sort of look at it directly through electrophoresis. So let's talk about restriction enzymes for this first technique called restriction digest. So here's DNA. That's what this molecule represents. Now, there are enzymes that are called restriction enzymes, and what they do is they, first of all, evolved as part of an ancient war, a war that's been going on for 3.5 billion years since the dawn of life, to protect bacteria from attack by phage. Phage are viruses that inject themselves into bacteria and destroy those bacteria. And that's happening at some unbelievable rate all over the planet. So what these restriction enzymes can do, these restriction enzymes are made by the bacteria, is they can find certain sequences that through evolution they've come to identify as viral sequences. And when they find those sequences, they cut the DNA at that point. If that were in the midst of a viral attack, that would short circuit the viral attack. So that's why it's done. So they find these restriction sites. That's what you see over here, this sequence. They cut the DNA at that point. And we can use this technique plus another technique called ligation to make recombinant DNA. So let's see what we have here. We have a segment of DNA that has a restriction sequence that's been identified at B. So at C, we have a restriction enzyme that finds that sequence and then makes a staggered cut. And in this case, it's called a sticky-ended cut. And the reason why is because you have nucleotides over here that have been broken away from their complementary partners. So you have an A that's sticking out there waiting to bind with a T, but there's no T over here. Here's another A waiting to bind with another T, a T that's waiting to bind with an A. So we've made the sticky-ended cut. Now, if we get another DNA fragment that has the right sticky ends, then all you have to do is mix them together. These actually work as liquids, and you sort of put them in the same tube. You mix them together, and often what will happen is that this fragment will find its way over here, and this will find its way over here. And so you'll have hydrogen bonds form. And then you need to do one more step. You need to add an enzyme called DNA ligase. And what it'll do is it'll seal this together with sugar phosphate bonds, making one DNA molecule. So that's how we make recombinant DNA, and that's what you see down here at letter J. Let's talk about what we can do now that we know about restriction enzymes. So the first part of this lab involves plasmid digestion. So what I want to point out is that the engineered plasmid that we're working with, P-ARA-R, has two restriction sites. One's called BAMH1, one's called HIND3. Those are named for the bacteria where they were first isolated. So BAMH1 makes a cut over here, and HIND3 makes a cut over here. So here's something I want you to think about. If we apply these two enzymes, how many fragments will result? How many pieces of DNA? And exactly how big will the fragments be? And you have two tips. You're told the length of this over here, and you're told the total length of the entire plasmid. Okay, so now what we're going to do is we're going to watch my colleague Johanna Berg go ahead and do a plasmid digestion. Let's see this in action. I'll talk you through the steps. So here's Johanna saying hello. She's adjusting the volume on the micropipette. She's putting on a sterile tip to avoid contamination or cross-contamination. She is taking the restriction enzyme. That's why I said R+. She's taking what looks like some buffer. That's creating the right pH environment for this restriction enzyme to work. She's now, it looks like she's adding the plasmid to that mix of restriction enzyme and buffer. Looks like she's removing it from the centrifuge. I must have missed that step. She's putting it in the hot water bath where it'll have ideal conditions. She's removing both. Okay, so one thing I want to emphasize is that everything we do twice. We're doing it once for our sort of experimental group. It's got the restriction enzyme, the buffer, and the plasmid, and then just the control, because we want to see what happens if we electrophoresis the plasmid without having broken it down with the restriction enzymes. And now she's spinning the whole thing down in the centrifuge, and there we go. Okay, so that's how you do a restriction digest. So if we make a cut over here with BAMH1 and we make another cut over here with HIND3, then what we'll do is we'll have one long fragment that goes from here all the way to here, and that will be 5,302 base pairs minus 807 base pairs for a total length of 4,495 base pairs. And then we'll have the shorter fragment that's 807 base pairs. Now in the plasmid setup that didn't receive any restriction enzyme, then we'll have this circle of DNA, the entire plasmid, that's 5,302 base pairs long. So what we're going to do now is set this up for electrophoresis. And let's see Johanna do this. Here we go. Okay, so the first step, what Johanna was doing is she was adding a little bit of loading dye. These liquids are clear, and if you want to get them into the wells of an electrophoresis chamber, you need to add some color to them. So that was the first step. So now she's taking the tube and she's got loading dye. Okay, so what's happening here is she's now in this sideways view adding some of our plasmid with enzyme to one of the three wells. And I actually don't know which one is which, but we've got three lines over here. Well, one of them is for a marker. This is DNA that's been cut to known lengths. One of them will be our plasmid plus restriction enzymes, and one of them will be our plasmid without restriction enzymes. So she's adding the third bit of plasmid DNA, and now here we see the gel actually running. It's pushing the DNA through the wells, away from the negative pole over here towards the positive pole over here. Now we're going to visualize it. And to visualize it, what we need to do is we actually need to put it in a special box that shines ultraviolet radiation on it, and that helps us visualize the DNA a lot better. And so we're putting it on there, and we're going to look inside, and this is what we see. Here's with restriction enzymes. We've taken our plasmid, we've cut it up into 2 fragments. Here's no restriction enzyme. It's of course going to be one piece because there was nothing to cut it up. And look at what we see over here. This lane over here shows a marker. This is DNA of known lengths. This over here is no plasmid over here. So this is something that we expect to be 5,302 base pairs. And then we have a fragment that you can see went a little bit further. Now, if it goes further, that means it's smaller, because a smaller piece will be able to make it through the gel more easily. So we're separating out by size. And one thing that I didn't tell you before is that the further you go, the smaller the fragment is. So we have one large fragment, one small fragment, and that's exactly what we'd expect to happen from this setup with these restriction enzymes. Over here, we just have one fragment whatsoever. Here we have a marker so that we can actually determine the length. If you knew the length of these, you could figure out the length of these. So I'm assuming that, like, for example, this band in the marker is probably about 800 base pairs, whereas this one is probably closer to 4,500 or something like that. So that is electrophoresis. Fantastic. We made it through the first part of this lab. Okay, so now we're going to move on to the second part of the lab, which is transformation. And what is transformation? We talked about this right up at the top of this video. Transformation is taking a plasmid and it's inserting it into a bacterial cell. So here's a bacterial cell with a plasmid. Here's one without. This is the main bacterial chromosome. Now, the first thing to know is that this is not easy. Plasmids are, as membranes go, large particles. In other words, they're not just going to diffuse through a phospholipid bilayer. They're also charged, because DNA has those phosphate groups, those negative charges, and that makes DNA extremely hydrophilic, and it's not going to pass through the hydrophobic membrane. So it requires some manipulation to make this happen. The first thing you have to do is you have to manipulate the cells. You treat the cells with a salt. It's called calcium chloride, CaCl2. That's called transformation solution. And then you do this procedure that's called heat shocking. And both of those vastly increase the probability that the plasmid that was engineered will be able to enter into the cells and then express the genes that we want to express. And let's take another look at this. So here we have DNA that's outside a bacterial cell, and it's not going to enter in. Here we have some that has entered in. Why? Well, the reasons are here. What the calcium chloride does, the transformation solution, is it weakens the membrane, and it also neutralizes the charge on the DNA, and that vastly increases the chance that the DNA will be able to enter. And the heat shocking promotes plasmid uptake, again, by disrupting the membrane, allowing the DNA, as it were, to slip through. So what we're going to do is we're going to see the process of heat shocking performed by Ms. Berg, and let's see her do it. So what she's doing now is, it looks like she's taking some nutrient broth, some yummy food that's going to help our bacteria grow. So over here in this styrofoam cup, just go back a little bit. In this styrofoam cup, there are bacteria that are competent, that are in these little microtubes. They were placed there we received these cells from Amgen. So we received competent cells from them. And then what Johanna just did was she fed them. One of these tubes is marked P-, one of them is marked P+, because we're going to put the plasmid in some of these cells. We're not going to put it in other cells. So let's see what happens next. What Johanna is now doing is aerating these bacteria. So just sort of mixing things up. Now Colleen's going to take over. Colleen's going to do the heat shocking. These bacteria are on ice. She's getting them all ready. They've been kept nice and cold. She's putting them in the warm water bath. And now she's removing them. Now that was vastly sped up. They actually stayed in that water bath for some specific period of time that maximizes the probability. So it's like you're sitting in ice water, you jump into a hot tub, and then you get back onto ice water. That's what we just did to those bacteria. That increases the chance that they're going to wind up being successfully performed. So now we're moving on to the second part of the transformation lab. We're going to plate out these bacterial colonies, and we're going to do a couple of other things. We're also going to screen them for antibiotic resistance, and we're going to turn on some genes. So let's talk about what's going on here. First of all, plating them out. We're going to grow these bacteria on petri dishes. These are widely used plastic dishes that have been used for 100 years. Well, they're probably glass before they became, they were sort of redesigned in plastic, but they're used for growing bacteria. On the petri dish, you put a culture of agar. Agar is the seaweed extract. We talked about it a little bit before, and then you can put food for the bacteria inside there. So that's why it's kind of this golden color as opposed to clear, which is just the lid. So now we're going to take our bacteria, that some of which we transformed, some of which we didn't, and we're going to put them on these plates. Now let's talk about the overall design of what we're doing in this lab. So here are 3 petri dishes. Every single one of them has an agar that's been infused with what's called lauria broth. Just think of a kind of beef broth. That's food for the bacteria. So all the bacteria will have something to eat, so it's possible that they'll all grow. Now, we're going to divide 2 of these plates in half. One side is going to get bacteria that were transformed with the plasmid, that's P+. One side will just get ones that were never transformed, those are P-. So one plate just has food for the bacteria to eat and for colonies to potentially grow. On the next plate, we've added to this lauria broth ampicillin. Ampicillin is an antibiotic. It's still in use by people today. If a kid gets an ear infection, they might take ampicillin to fight that off. Half of the plate will get P+ bacteria, ones with the plasmid. Half of the bacteria will get P- ones without the bacteria. And then finally, we have a third plate. This third plate has lauria broth, the food. It has ampicillin, the antibiotic, and it also has a substance called arabinose. And arabinose is an inducer for the arabinose operon. Basically, it turns on the gene that allows expression of the red fluorescent protein. And we'll talk about how that is in a little bit, but for right now I want you to pause and make some predictions. So here, 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 and here. Will the bacteria grow? The second question is, if they grow, will they be red and will they glow under ultraviolet light? In other words, will they express this red fluorescent protein? So I want you to make a prediction, and then I want you to justify that prediction, and then we'll see what happens. Okay, so now what we're going to have is Ms. Berg is going to carry out some of the steps involved in this transformation. She's going to plate these bacteria and she's going to incubate them. I'll talk you through it. So what she is doing now is taking these bacteria, and these are the bacteria that we had just heat shocked. And so now you can see she's got this petri dish over here. And what she's going to do is, using the micropipette, she's going to place the liquid onto the surface of the nutrient agar. Now she's going to use a spreader to spread out the bacteria all over the surface. And so that looks like these are all the P- bacteria. So she's going to do the P- on this plate and then the other plate. And now this is the plate that has the LB and the ampicillin and the arabinose. So she's putting P plus on that. And let's see what she's going to do next. She's going to take the spreader. I've got her sped up here, and she's spreading this out. She's spreading out the P plus on all 3 plates. So spreading out the P plus, and then she's doing it again. So now she's taking these plates where she's has the bacteria, and she's going to put them in the incubator, and she is remembering that you've got to put them in upside down, otherwise they'll be super messy. Getting a big thumbs up. Way to go, Johanna. We've now incubated our bacteria. We let them grow, for about 24 hours. Let's see what happens next. Holy cow! These are our results. So let's look at what we've got here. So this was just bacteria food, the Loria broth. Over here, the P plus bacteria have grown and they've produced a lawn of bacteria. Over here, the P minus ones, the ones without the plasmid, did exactly the same. Why is that? Okay, so now over here, this is the P plus side. This is LB and ampicillin. So we've got bacterial colonies. Each one of these represents a single bacterial cell that then grew into several million clones. And so we've got, hundreds of bacterial colonies on this side of the plate. On this side, where we place the P minus bacteria, nothing is growing whatsoever. Now on the third plate, this was LB, ampicillin, and arabinose. And note how the colonies are growing here, but they're also quite different. So these are showing a red pigment. So what I want you to do is I'd like you to pause, explain what happened. Why do we have these differences by these in these 3 plates? Okay, so now what we're going to do is we're going to hear my colleague, Sydney Ardall, explain what's going on. Let's hear it from Sydney.
All right, team, time to look at some results. So the first one that we're going to take a look at is our LB plate. So this plate, you can see that we had growth of both the plasmid bacteria and the non-plasmid bacteria. So on this plate, we had growth of both of them. This is the LB plate. Up next, we're going to talk about the LB amp plate. So this plate had ampicillin inside of the gel. And you can see that we only had growth of our bacteria on the P positive, or the bacteria that had the plasmid in it. On this side, we did not have the plasmid that had the ampicillin resistance, and therefore these bacteria did not grow. Finally, we have our LB amp era. So we had that arabinose as well as ampicillin resistance ampicillin inside of this gel. And so you can see that our plasma is going to grow. And if I get closer, you can see that there is both pink and white colonies there. Those pink colonies are going to be the ones that glow.
So conceptually, the hardest thing to understand in this lab is what's going on with the arabinose. There's a connection to gene regulation, and there's a connection to operon. So this is a fantastic opportunity for us to review some important AP biology content as we understand this lab. So arabinose is a 5-carbon sugar. Remember, in these kind of structural formulas, there's a sugar and unseen hydrogens at every angle vertex over here. So 1, 2, 3, 4, 5 carbons. So here's how the arabinose operon works. If you remember back to the lactose operon, it's pretty much the same mechanism. So we have a regulatory gene over here that produces mRNA that gets translated into a regulatory protein. This regulatory protein is capable of binding on the other side of the promoter for the gene, and therefore it'll block RNA polymerase from transcribing the structural genes. In other words, when there's no arabinose, the cell doesn't make enzymes to digest arabinose. But when arabinose is in the environment, what the arabinose does, that's what this represents over here, the arabinose binds with this regulatory protein. That causes an allosteric shift in the structure of the protein where the binding sites change shape. And when they change shape, they no longer bind with the operator. And because of that, RNA polymerase, after binding with the promoter region, can go ahead and transcribe all of the genes. And these genes make a series of enzymes that digest arabinose. Fantastic, that's what exists in nature. Well, what we have in this lab is a little bit different because in the P-ara-R plasmid, what's happened is that the structural genes for arabinose digestion have been removed and they've been replaced with the red fluorescent protein genes. So instead of this system over here that creates these enzymes for digesting arabinose, what we have is this. When there's arabinose in the environment, it'll bind with this arabinose regulatory protein. It'll cause this allosteric change. It won't be able to bind with the operon. RNA polymerase will ride down the length of the plasmid and it'll transcribe the RNA for red fluorescent protein. So it'll transcribe this gene, it'll make mRNA for red fluorescent protein, and that by a ribosome will then be translated into the red fluorescent protein that's causing the change. So having heard that, you should be able to explain now the entire thing. So what's the difference between this plate where the cells are growing but not expressing RFP, and this plate which has LB and ampicillin and arabinose? They both have LB and ampicillin. Here they have LB, ampicillin, and arabinose, and here they're expressing all three. So, and here's actually a video where we can sort of see the difference.
All right, as you can see here, we have those pink colonies that put under this light are able to glow. So we had a successful transformation.
And now you can see those colonies and all their shimmering fluorescent beauty. So again, the goal for you as an AP biology student is to be able to explain the difference between why the colonies aren't glowing over here in LB and ampicillin and why they are growing over here in LB, ampicillin, and arabinose. The operon is right here. Your job is to pull it together. I want to thank my fantastic colleagues at Berkeley High School, Ms. Colleen Simon O'Neill, Ms. Johanna Berg, Ms. Sydney Ardal. You're the best. I'm so lucky to be working with all of you. And thanks for doing all the hard work in pulling together all the pieces for this lab. I also, of course, want to thank the folks over at Amgen because they provided us with the materials so that we could do this as a demonstration for our students. And, you know, next year we expect our students will actually be doing this lab. So folks at Amgen, thank you very, very much. So again, thanks to all of you. Go to sciencemusicvideos.com or learn-biology.com to learn more. And to end, I can't help but show you this.
Oh, that's a good pupper. Come on. Let's go to the lab. Come on. Oh, nice job.
Guys, thank you so much. Leave me comments. Let me know if you have any questions. Go to learn-biology.com, sciencemusicvideos.com. Really appreciate your being with me. Thank you very much.